This is the story of an enzyme engineering project in the AI era. 195x improvement in productivity, 12x improvement in specificity, 7 generations in 16 months, and this enzyme just kept getting better. So a pharma company came to Ginkgo looking for a hydroxylase. The goal was to improve the manufacturing process for a particular pharmaceutical ingredient to make it faster, cheaper, greener. They wanted to replace a chemical hydroxylation step, and they had identified enzymes that could do similar reactions. The challenge was that this particular substrate was about twice as large, and the known enzymes weren't getting the job done. They needed more productivity, an enzyme that converts most of the substrate in 24 hours. And they needed high cell activity, meaning it makes the desired product with the right chirality and not the stereoisomer or any of the possible position isomers. Easy, right? I mean, probably would have been impossible 10 years ago, but times change. So how did we do it? Step 1, assay development. You can't improve an enzyme much without real performance data. For the primary screen, we put together a high-throughput assay called EchoMS. This only takes about 1 second per sample, so it can check thousands of enzyme designs per run. It can measure the product we're looking for and separate it from most, but not all, of the byproducts. For the secondary screen, we developed a long LCMS method capable of separating exactly the desired product from the 5 possible isomers. So we could cherry pick the top performing enzymes and make sure they were working exactly as expected. Step 2, metagenomic discovery. We started with 3 seed sequences of enzymes known to do chemistry similar to what we wanted. You do a simple blast search on those and you get 183,000 candidates. That's too many to test, even for us, so we had to narrow it down to a tight 1,000. We apply 2 main kinds of filters here: structure-based and sequence-based. Structure-based means 3D models and molecular dynamics. We dock the substrate against the active site to see if it fits with the residues associated with catalysis. Sequence-based means protein LLMs and other AI models. These will assign a likelihood score to a particular protein sequence that tends to predict desirable properties like structural stability or catalytic activity. We use AI embeddings to cluster candidates by similarity, ensuring that we don't waste time resampling enzymes that are functionally the same. Step 3, build and test. Here, I'm showing all the natural enzymes we considered. Dots that are closer together have more similar sequences. The blue dots are the ones that we physically assembled the DNA for in the Ginkgo Foundry, running them through the assays from step 1. These 2 are the top performers. When I was in lab, there was essentially no way to computationally filter through 183,000 candidate sequences to find a desired activity. These days, we can do it pretty consistently. Step 4, protein engineering. Starting from these 2 natural enzymes, we made targeted sequence modifications to improve the performance. We use an ensemble approach, meaning we'll try multiple strategies using our best guesses about what works for a particular enzyme. We use structure-based tools to target the residues that control docking in the active site. We use sequence-based LLMs to generate new amino acids that resemble the products of natural evolution. The result was a large library of sequences, about 2,100 total, that we could screen for activity with EchoMS. Starting from one of the natural sequences, we found a variant with 4x better productivity. The other natural sequence gave us a variant with 2x better cell activity. Unfortunately, these improvements were not together in the same enzyme, but we're getting there. Because step 5 is iterate with assay-labeled data. All the sequences that we've generated, all the experimental measurements we've collected, these are now inputs for a problem-specific AI model. Our software stack, called OWL, can use all of this data to predict productivity and selectivity, then generate a new set of enzymes from everything we've learned. This is the real power of the Ginkgo Foundry. We can generate training data quickly. For this project, 1 iteration was less than 2 months. More enzyme sequences, more tests, more data, better models. Generation 2 got us 34x higher productivity. Gen 3 was 73x, 100x, 129x. By Gen 7, we're at 195x productivity, 12x better selectivity, and as far as we can tell, no compromise to enzyme expression level or stability. I have never seen anything like this. In my lab days, I might have stopped at Gen 4 thinking 73x must be pretty close to the limit of what's possible. But I've never worked on a bio-AI project powered by this much high-quality data. This thing just kept getting better. Now, these are results from just one enzyme. It doesn't mean every project will go this way. But we went from discovering a catalytic activity that was new to nature to an enzyme with this level of performance in about 18 months. Part of this is made possible thanks to the big foundational AI models that everyone's talking about: AlphaFold for structural modeling, ESM2 for sequence modeling. That gets you to Gen 1. Going from Gen 1 to Gen 7 took about 10,000 data points, tracking productivity and specificity for carefully selected real enzyme sequences. This was not zero-shot learning. AI is data hungry, and if you want to see what it looks like at peak performance, you have to feed it real biological measurements. So if you've ever thought about using more enzymes in your pharmaceutical manufacturing process, the space of possibilities is opening up. If you were amazed by AlphaFold and ESM2 when they came out a few years ago, the space of possibilities is opening up again because the combination of AI and high quality data is taking enzymes somewhere new. And we've never been here before.